Hi everybody. So this is lecture 1 of the renal physiology portion of physiology. We're going to have another 2 lectures. So this is actually an important lecture for you guys to review as the next lecture that will be in class will be picking up from where we left off. So first let's just start by reviewing the functions of the kidney. We understand that the kidney filters our blood and it excretes wastes. We're going to talk obviously more to that in this lecture and the next lecture. We also hopefully would recognize that the kidney is involved in helping us regulate blood pressure because it can help us regulate our blood volume, right? And we know that volume is related to pressure. It can also help us regulate our osmolarity. And what's interesting about that is the kidney's ability to separate water versus solute when it comes to reabsorption and excretion. So it can actually separate the solute from the water and reabsorb that, or just reabsorb the water and get rid of the solute. And by being able to separate the two components, it can adjust for osmolarity. So it regulates electrolytes, which we'll talk about, like sodium chloride, and potassium, and calcium, and so on, and acid-base balance. So retaining or getting rid of hydrogen ions, bicarbonate, and we'll discuss more to that in the third lecture. It also secretes hormones like erythropoietin in states of hypoxia, which helps stimulate red blood cell production. It also helps regulate our calcium by participating in calcitriol synthesis. So we'll talk more about that in our calcium lecture. It can clear hormones from the blood. And then in starvation, interestingly, they can actually synthesize glucose from amino acids, which is a process referred to as gluconeogenesis, or can partake in that as well. We commonly think of the liver when we think of this, but the kidney can also do that. So just the basics of anatomy and the urinary system. There are essentially 6 components to this. You have the 2 kidneys, okay? So we have 2 kidneys, one on either side, right? Retroperitoneal structures, and you see that here at the hilum, where there is the arteries and the veins entering and exiting. You also see this structure here called a ureter. So you have 2 ureters. The ureters descend into the pelvis where it connects with the bladder. And then the bladder, of course, can contain and store urine. And then ultimately the urethra, where it would be exiting finally. This is just a slide of a cadaver, and you can see the liver here has been cut away, or at least partially sectioned, so that you can get a better view of this structure here. Now that structure is the inferior vena cava, and you'll notice at the hilum of the kidney on each side, you can see the inferior vena cava, the renal vein connects directly to the inferior vena cava on either side. And I'm pointing this out because I want you to understand that the kidneys do not connect to the portal vein. The portal vein, which we'll get to more in GI, is actually the venous system that drains from the digestive tract, so it can be filtered through the liver first. So the kidneys does not drain into that portal system, it drains directly into that inferior vena cava. You can also see the abdominal aorta here, okay? You can see where it bifurcates here at the lower end, right? It's the common iliacs. And you'll see up here, just a little bit behind the vein there, you can see the renal artery. So the renal artery comes right off of the abdominal aorta into the hilum there. And over here, you can actually follow and see the ureters descending towards the pelvis there, okay? Okay, so on this slide here, which has a cartoon of the kidney, you can see it's been sectioned open, and what you see on the outside here is called the fibrous renal capsule. So it's a fibrous, stiff structure. It helps give it its rigidity and strength and the shape. And then just underneath the capsule, you have the renal cortex. Okay, so that's underneath the capsule as you follow that around here. All right, that's the cortex. Now the cortex, just beneath that, or just deep to that structure, you actually have the medulla. Right? So we've heard of this before, cortex and medulla. The cortex are usually on the outside, and the medulla is a deeper structure within. And so this medulla, or medullary region here, contains these triangular looking structures, which are referred to as pyramids. So those are referred to as the renal pyramids. And each kidney has several of them. So you can see here 1, 2, 3, 4, 5, 6, 7 in this particular cartoon. It could be 7 up to maybe 9 or 10 or so. And these pyramids are important structures that we'll talk more to as we continue. But here again, the pyramid's located in the medulla. You'll notice that the apex of the pyramid really faces downward here. So this is the apex down here. It actually contains a lot of little pores there that kind of look like a little nipple at the very end. And so it's referred to as a renal papilla. And you'll notice that the tip of that pyramid actually is surrounded by this connective tissue referred to as a calyx. And the calyx kind of looks like a cup. And so it's kind of like a cup at the very end of that pyramid there. And it's ultimately going to be catching urine as it's being processed through the pyramid. And we'll talk more about the anatomy and the structure and what's going on there. But understand that urine will be being produced starting in the cortex, moving into the medulla, into this pyramid-like structure. And then ultimately, as it's being formed and ready to be excreted, it drips into the calyx. And these calyces, we call them minor calyces. They're the ones that connect directly to the pyramids. They coalesce to form major calyces. So we have several major calyces. And then those calyces coalesce to form the renal pelvis. And so you can see that right here. And the renal pelvis ultimately becomes the ureter. And the ureter leads to the bladder, right? A couple of things to note here on the cartoon is that in between each one of these pyramids here is called a renal column. So that's another term here. In that renal column, you're going to see things like blood vessels traversing it. So it's an important structure to get the blood vessels up towards the cortex. So the blood vessels won't technically travel through the pyramids, but through those columns. In addition, we have another term, which is not labeled on here, unfortunately, but it's the pyramid plus the cortex that's above the pyramid. So that's two things, the pyramid and then the cortex, which is located just above each pyramid. So that pyramid and its cortex is referred to as a lobe. So that would be the lobe of the kidney would be the cortex and the pyramid that go together. All right. And then finally, there's something called the renal sinus. So if you look here at the hilum, all right, where the arteries are entering into the kidney and you see the veins are leaving, it enters into sort of a little opening space there, referred to as the hilum. Excuse me, as the renal sinus. And you see the ureteral structures here with the calyces. This could be filled with, like, say, some adipose tissues. Some nerves might be entering there as well. All right, so let's see the next slide. So here you can actually see the anatomy on an actual kidney. Highlighted in green would be the region that's being labeled. So papilla would be at the end of the pyramids, and as you can see here, this little sort of smooth region in the medullary portion of the kidney, and at the very tip of that, which would be the apex of that pyramid, is the papilla. And then the sinuses, that little area in the hilum I was speaking about. And again, you'll see some sort of connective and fatty tissue in there, but that's also the region where arteries and veins are entering and exiting, again where the calyces are coalescing to form the ureter, and where the nerves would be entering. The minor calyces are labeled over here, which you can see just underneath the pyramids. They coalesce to form major calyces, and the major calyces coalesce to form the renal pelvis, which you can see down here, and then finally that becomes the ureter. Okay, so that's the basic anatomy of the kidney. But the kidneys themselves are actually composed of millions of functional units referred to as a nephron. In fact, each kidney has approximately 1.2 million nephrons, for a total of about 2.5 million. And the nephrons are actually where really the sort of the business end of the kidney is taking place. This is where filtration of the blood occurs, and ultimately where sorting out what's waste and what needs to be conserved is going to occur. So there's really two principal parts. You have something called the renal corpuscle, and the renal corpuscle is actually where the blood is filtered. So that's gonna be how the blood enters into a nephron. And the nephron is really essentially a hollow tube lined by very specialized cells, and these cells, their job is to reabsorb and to secrete, and ultimately to form the urine. Right? So these renal tubules are very long, they're coiled, they have these specialized cells to get that job done. So let's just talk first about where the filtration is going to take place, how the blood actually gets into the nephron. And so that's going to occur in the renal corpuscle, and it consists of really two components, which is the glomerulus and something referred to as the Bowman's capsule. And the Bowman's capsule is really a membrane that surrounds or engulfs a specialized sort of capillary of the blood vessels. And so let's talk about that for a second. Okay, so let's just take a quick look at what a basic structure of the nephron, and I just want to show you that corpuscle that I was referring to. So let me see if I can get this here for you. All right. And we'll use blue for now. How about that? So over here, this region is the region I want to bring your attention to right now. This right here. All right. So that region that I just circled for you, that's the corpuscle. All right. What it really consists of is in black right here, which you can see this black portion here. All right. And you can see this sort of curvy tubule here. That's the nephron. Okay. Kind of looks like a strange looking snake. Now, over here in red, all right, this is actually the artery. In fact, this red over here, I want to label that as the afferent. Sorry about the handwriting. Afferent arterial. Handwriting is atrocious on this thing, so I apologize. And then down here, this portion of the artery here is the efferent arterial. Okay, now you have the afferent arterial, which is again where blood is coming into, or is going to be coming into this specialized capillary here. So that's where blood is coming through here. It's going to enter into this capillary. So that's why we call it afferent, because it's going towards it. This capillary in red, all right, it kind of looks like a little fist that I drew here. That is called the glomerulus. Okay, so that's a special term. That's the glomerulus. That's referring to the capillary. And then some of that is going to get filtered. All right, so we're going to have some filtration taking place here. Excuse me. All right, so now we have over here, some of that blood is going to get filtered and go directly into this hollow tubule system here. All right, so you can get blood that's going to be moving through this entire tubule system. All right, down to this loop. All right, through the loop, ultimately to end up over here into something called a collecting duct, which I'll refer to in just a second, or I'll talk about more in a little bit. And then some of this blood that came from the afferent arterial, not all of it's going to get filtered, so some of it's going to actually make its way out through the efferent arterial. So you might notice something different about this capillary. First off, is that unlike the capillaries we talked about in the past, where there is an arterial, a capillary, and then a venule, in this case, what we're talking about now is a afferent arterial, a capillary, and an efferent arterial. So this capillary system is embedded between two arterial systems. All right. So that also makes it fairly special. And these arterioles, as you guys know, have a lot of smooth muscles, which allows them to dilate or constrict to a greater capacity than, say, a vein would. So that's going to be an important thing for us to talk about in a little bit. Now, first off, when you take a look at this capsule, okay, you can see this in black, the capsule. That would be the Bowman's capsule. And the Bowman capsule tightly adheres to the capillary surface, so that would be the visceral portion of it. Kind of like when we talked about the visceral pleura or the parietal pleura, the visceral pericardium or the parietal pericardium. It's similar to that in that you have a visceral layer which is adhered directly to the surface, and then you have a parietal layer which is in the outer surface there. Again, kind of like a fist into a balloon. And there, the blood will then be filtered into the space between the visceral and parietal layers, and it enters into that space. Once it's in that space, it can move into the next segment of the nephron. Okay, and that next segment is known as the proximal convoluted tubule, sometimes just referred to as the PCT. Again, proximal because it's proximal to the Bowman's capsule, convoluted, all right, and it's a tubule. And then it dips down into this little loop right here, which sometimes just referred to as a nephron loop or a loop of Henle, or LOH. So it goes through the loop, the descending portion that ascends, comes back to another proximal tubule, but this time this one's distal, so it's called the distal convoluted tubule. or the DCT. And the distal convoluted tubule connects to this large tubule, which is referred to as a collecting duct. And you'll notice that I also drew some other things entering into this collecting duct. So you can see right there, over here as well, another one over here. Ultimately, what that's showing you is this. This collecting duct collects blood or filtrate from multiple nephrons. So for example, in the kidney, you could have, say, 250 of these collecting ducts in total, and each collecting duct will connect to about 4,000 of these nephrons. All right, so this is essentially where a lot of nephrons funnel to, is this collecting duct. All right, and then the last thing I want to point out in this drawing here is that I have a dotted line or a dashed line right here, all right, which you can kind of see me kind of cutting off the loop there. And that's actually to delineate the fact that this proximal convoluted tubule and distal convoluted tubule are located in the renal cortex, And this line, once you pass through this line here, we are in the medulla. All right, so that's just to help us anatomically see how this nephron sits within the kidney. Now, this cartoon, I've just gotten rid of some of the details here, but I've also drawn in another piece that I want you guys to see, and that is here. Just use green again. And that's to show you that over here, again, this was our afferent, all right, arterial, and then over here was the efferent on the other side, right? So that's our efferent arterial. And so blood was coming in this way into our glomerulus, into this whole thing with the glomerulus and the Bowman's capsule is referred to as a corpuscle. All right, so it's heading into our glomerulus, some of which is going to get filtered into our tubule, and then the rest of it is going to enter into the efferent arterial. Once it goes into the efferent arterial, now this is blood that is not in the nephron. This is what did not get filtered into it. So this blood will then actually make its way into a capillary system that surrounds the nephron. So it closely surrounds the proximal convoluted tubule, the distal convoluted tubule, and also dips down here, which you can see, I'll draw a little arrow into it, dips down here and is closely associated with that loop of Henle. And now this is like a typical capillary where you have arterioles, a capillary, and then over here in blue you can see this is where all the blood will be draining to. So this would be sort of our venules, ultimately to return to veins over here that leave the kidney. So that would be more of a typical capillary setup as opposed to the one that we saw with the glomerulus. Now this capillary system again follows the general rules that we've talked about before in terms of filtration and reabsorption, with an arterial on one side and a venule on the other. And so these are in close proximity to all the special cells of the nephrons. In this way they can do exchanges with the cells of the nephron. So the cells that line this nephron, they'll be reabsorbing or secreting, and so when they reabsorb, it can get brought back to the blood into these capillaries, or if the capillaries have other substances that they can secrete, they'll secrete them into the lumen of the nephron. So just because blood didn't get filtered at the very beginning in the corpuscle doesn't mean that some of the stuff that's in that blood that makes it into the efferent and ultimately into the capillaries, it can't get back into the nephron. So we'll talk about that. So in other words, we can have some kind of toxin that may have made it, somehow over to here, all right, that can still make it into a nephron. It could still get filtered, or excuse me, I should say, still get secreted into that nephron ultimately. And so we'll talk about some of how that happens. Okay, so here again, this is the cartoon of what we've already seen, which is the nephron. And you can see here, this is the corpuscle, which has the glomerular capsule or the Bowman's capsule, and the glomerulus, which is actually the capillaries. The afferent arterial, which is where the blood is entering, to be some of it to which be filtered, and then the rest of the blood will leave through the efferent arterial. And the blood that does get filtered here in the corpuscle will enter into the proximal convoluted tubule through the loop of Henle, and then into the distal convoluted tubule where it will connect with the collecting duct. What I like about this cartoon is that if you look at the top here where you can see a slice of the kidney, where you can see the lobe of the kidney. Here's the pyramid, and you can see the cortex above it. You'll see that this nephron, the corpuscle, the proximal and the distal convoluted tubules are located in the cortex, not in the pyramid. Okay, the loop of Henle dips into the pyramid, and that's actually an important distinction. That's why I drew that little dashed line in the cartoon in the previous slides. And then you'll see that the distal convoluted tubule connects with the collecting duct in the cortex. Then the collecting duct does go and dip into the pyramid and ultimately empties into those papilla, the renal papilla there, which are going to be excreting or dripping the processed now urine into the calyx, into those minor calyces. Now, in terms of how the blood that's supposed to get filtered reaches the nephrons, and if you recall, the nephron, the glomerulus, and the, or I should say the corpuscles, they're located in the cortex of the kidney, and blood enters through the hilum. So it has to go into the hilum, all right, and the hilum's right over here, the central region here, has to enter, and then it's going to branch, and those branches are going to ascend to the cortex. So it's kind of important to understand how the blood gets there. So again, the arterial blood is coming from the aorta, so the renal arteries, a branch off the abdominal aorta, that enters into the hilum, and as soon as it enters into the hilum, it branches into what we call a segmental artery, so different segments of the kidney. And then those will actually further branch into interlobar arteries. If you guys recall, the lobe of the kidney is the cortex and the medulla, the pyramid and the cortex located above it. And so it goes between each of those lobes, all right, through the column. It doesn't go through the pyramid, it ascends through the column, or goes between those pyramids in that column space. And then what you'll see is that it hooks over the top of the medulla, so where the pyramid, the top of the pyramid, or I should say the base of the pyramid there, meets the cortex. You'll see that the blood vessels take a turn, like almost a 90-degree turn there, and essentially rides between the cortex and the medulla at that region. All right, and that's called the arcuate because it arcs over it. And then you see the cortical radiates, meaning that they radiate out into the cortex. And those cortical radiates will ultimately turn into the afferent arterioles. They'll branch into those. The afferent arteriole will connect with the glomerulus, which will then have the efferent arteriole. Again, we're still arterial. The efferent arteriole will then turn into the peritubular capillaries, which is the capillary system that surrounds the tubules, and then the vasa recta, which are the capillaries that follow the loop of Henle. So we give them different names, even though they're still just capillaries. Okay, and then ultimately those capillaries will coalesce into venules, the cortical radiate veins, the arcuate veins, interlobar veins, and ultimately back out to the inferior vena cava. And at the bottom here, I pointed out that the kidney receives 22% of the cardiac output. Okay, and if we're going to use our average male, who has about a cardiac output of about 5 liters. Okay, so just to point this out here. So if our average male has a cardiac output of about 5 liters per minute, all right, that's the cardiac output. And 22% of that, all right, so what's going to the kidney, 22% of that is going to be 1,100 mils, or 1.1 liter, okay, per minute. So it gets a very large amount of volume every minute, or a very large chunk of the total cardiac output. Okay, so in this way, it can filter a lot of our blood fairly regularly in order to maintain good homeostasis. Okay, so here's a cartoon looking solely at the corpuscle, which again is the glomerulus surrounded by the Bowman's capsule. And remember I said that the Bowman's capsule has 2 layers. It has a visceral layer which directly adheres to the glomerulus, and then there is the parietal layer, which is the outside layer here, and then there's that space. So when blood goes in through the afferent arteriole, it gets filtered into that space. The blood that doesn't get filtered goes out through the efferent arteriole, and the blood that was filtered ends up going into the proximal convoluted tubule, which you can't really see here, so it can be cut off. All right. One other thing I wanted to point out here is on the visceral layer okay, the Bowman's capsule, the one that's directly adhered to the glomerulus, has these specialized cells called podocytes. So these podocytes have these sort of slit-like interdigitations here, which you can kind of see. And there's a little bit of space there, which helps to further sort of filter the blood. So I like this cartoon because what it's showing you here is that ultimately at the corpuscle, you can see we have two nephrons you can see here and one collecting duct. So these two nephrons are sharing this collecting duct here. So here you can see the corpuscle, over here you can see the corpuscle. They can be located very close to the medulla, so that's why they call it juxtamedullary nephron, or it can be located further up into the cortex, so a cortical nephron. Ultimately, we don't need to know the difference at this point, but just a couple things I wanted to show you is that here, this corpuscle, they have the afferent entering into the to form the glomerulus, then the efferent, and the efferent actually branches into this vast capillary network, which would be our arterioles turning into capillaries, and then the blue, you can see the venules, and they closely surround the tubules of the nephron, again, so that exchange can continue to happen between the nephron and the blood. Now we have a different name, but it's ultimately the same thing, where the efferent arteriole will branch and form capillary network that surrounds the loop of Henle. Ultimately, conceptually, it's the same idea. We have a capillary network that helps us to exchange with the loop of Henle, and we refer to this capillary network as the vasa recta. So the vasa recta and the peritubular capillaries. So this cartoon is just solely to just give you guys an idea of the basic stages that we talk about when we filter blood and ultimately form urine. So the very first stage here at the top, called glomerular filtration, which is what I've been speaking about, right? Afferent, efferent, and it comes into the glomerulus, blood is filtered, so a fraction of that gets filtered into this nephron. And that's the only thing that happens here in the corpuscle, only filtration. Okay, there's no reabsorption, there's no secretion, it's simply filtration. Once it's filtered and gets into the proximal convoluted tubule, now we're in the tubular system, and we can have tubular reabsorption or secretion. Reabsorption is simply taking whatever got into the nephron and removing it and reabsorbing it back into the bloodstream. Okay, and in fact, that's the majority of what the kidney does, is reabsorb things. Okay, now again, remembering that this is to represent the capillary system that surrounds the tubules, so it gets reabsorbed, and since the capillaries are right there, it goes right back into the blood. Tubular secretion, on the other hand, is going to be components of the blood in that capillary that are still able to secrete and move substances into the tubule lumen. So we're having exchange occurring. We're taking stuff out of the tubule lumen and we're putting stuff into the tubule lumen. And so we're going to have specialized cells that allow us to do this. And we'll talk about that more later. And then at the bottom here, it's talking about water conservation, where ultimately, towards the end of the tubule system, we can kind of fine-tune or adjust our water conservation or reabsorption versus excretion. And we'll get into that more later. But the final process is really producing urine here, which you can see these little drips over here. And that's referred to as excretion. Excretion is waste. It's basically getting rid of, this is what's going to be urine. This is leaving the body ultimately. Okay, and that's different from secretion. All right, excretion and secretion are not the same thing. All right, but just keep in mind, excretion is ultimately the waste, the urine that we're going to get rid of. Okay, so this is a cartoon that you guys have in your notes, and it has a couple of things in it that are important to know and understand. So I want to go through that with you right now. So first thing is just to kind of orient you guys that this is the afferent arteriole. All right, and you can see here the divide where some of that blood that's making it to the nephron is being filtered into the tubule lumens. So this would be this region right here would be where the filtration is taking place. That would be the corpuscle. That's the Bowman's capsule and the glomerulus. And then that enters into proximal convoluted tubule, down the loop of Henle and up it, and then distal convoluted tubule into the collecting ducts, ultimately to produce down here our urine. Okay. Now, not all the blood that goes to the kidney or to the nephron here or into the glomerulus is actually going to get into the nephron. In fact, a large portion of it does not, and it goes into the peritubular capillary system instead. So that would be what this represents, the peritubular capillary system, as well as the vasa recta, which surrounds the loop of Henle. Okay, and ultimately this blood goes back into systemic circulation. All right. Now, a couple of numbers that we need to understand is that, first off, looking at both kidneys as a whole. So when I'm talking right now and giving you examples and just showing you a single nephron, understand that that single nephron represents the entire population of nephrons of both kidneys. So the volumes I'm talking about are referring to total volumes for both kidneys, not a volume for one nephron. Okay, this is volumes of total volumes of all the nephrons collectively. All right, so that volume, again, for both kidneys collectively, is going to be 1,100 mils, all right, per minute. If you guys recall from previous slides, the 1,100 mils is actually 22% of the cardiac output. And so that's how much blood flow is going to the kidneys every minute, okay? And then this way, it makes it so that the kidney can constantly be filtering blood and helping us to maintain our homeostasis and get rid of toxins. So a couple things. That 1,100 mils, that's whole blood. Just as a reminder, whole blood includes the cellular component, which is the red blood cells, white blood cells, and platelets, as well as the plasma. So if we're talking about the cellular components of this volume, that's equivalent to about 500 mils per minute. And what's important to know is that 500 mils, again, this is for the cells, that's the cellular volume, that doesn't get filtered when it reaches the corpuscle in this region over here. In the glomerulus, it doesn't get filtered. Those structures are too large. So just like capillaries we've seen before, red blood cells, white blood cells, platelets, they cannot get through the filtration there. All right, which leaves the last volume, which is 600 mils per minute. That is going to be equal to the plasma. The plasma portion, all right, which is more sort of the fluid portion of it, which has all our dissolved electrolytes and so on. That makes about 600 of that total 1,100. And so that 600 can be filtered, all right, or has the ability to be filtered. It's not all going to be filtered. It's not going to filter 600 mils per minute. So what happens is it's going to take a fraction of that 600 and it'll filter it. Okay, so of the total 1,100 mils of whole blood, 600 mils of it is capable of being filtered. Okay, the reality is, of that 600 mils per minute that can be filtered, only 20% of the 600 can be filtered. And that comes out to, in this case, 120 mils per minute. In other words, of the 1,100 mils per minute of whole blood, 120 mils of plasma, 120 mils per minute of plasma actually gets filtered and enters into the nephron over here. So what actually comes in is 120 mils. Okay? Again, that's over time, so that's per minute. So 120 mils per minute enters in there. All right? Now, that plasma that enters or gets filtered into this nephron tubule is essentially exactly like the plasma of the blood, with one very key exception. It doesn't really contain any proteins. So the plasma that gets filtered into the nephron has no proteins. Sorry about that. This should say proteins. Okay, so that does not get filtered in. They're too large and usually negatively charged, and they get excluded. So ultimately, the plasma that gets filtered is like the plasma of the blood without the proteins. So everything that gets excluded are the cells, red blood cells, white blood cells, platelets, as well as the proteins. Okay, and so that plasma that does get filtered, typically that volume is about 120 mils per minute. And in fact, what we call that filtrate that enters into the nephron, what we call that rate or volume rate, is called a GFR, or glomerular filtration rate. And in this case, under normal circumstances, it's typically anywhere from about 100 to about 125 mils per minute. Okay, so that's our typical GFR. And so if you look at some of this, what's written out here, you can see he goes into talking about the 20% of the plasma here. Okay, that's the GFR. In this case, it was about 120. All right, and once that enters in here and it's been filtered, several things happen to the fluid inside that lumen. One, it can actually be reabsorbed, which are these black arrows here, going back into the capillaries. So a lot of reabsorption is going to take place. At the same time, some of the stuff that didn't get filtered initially can be secreted in. So we refer to that as reabsorption and secretion, and it's ultimately just exchange between the capillaries, the blood capillaries, and the nephron lumen. There's going to be so much reabsorption occurring that by the time we get to the very end, usually we have reabsorbed greater than 99% of whatever got filtered in via a lot of reabsorption, which is why these black arrows are a little bit larger than the secretions inward. And we produce a very sort of usually concentrated urine, or a very much lower volume urine. Okay. So urine formation. So GFR, or the glomerular filtration. So we talked about filtration occurring at that corpuscle. Now it gets into the nephrons. So with that 120 mils per minute entering into the nephrons collectively. The kidneys are going to convert that plasma, all right, to urine by basically first filtering. And by filtering, we've excluded proteins and blood cells. Then tubular reabsorption. We're going to reabsorb, and that's primarily what's going to happen. And then we're going to have tubular secretion, which means we can still move stuff from the blood in the peritubular capillaries into the nephron tubules, and then ultimately excretion. Okay, the glomerular filtrates, all right, again, similar to blood plasma, except, again, no proteins. I want to reiterate that point again, is there's no protein, okay? Tubular fluids from the proximal convoluted tubule through the distal, substances are removed or added. Again, that's going to be secretion versus reabsorption. And then the urine that finally enters the collecting duct, really by the time it reaches the collecting duct, there's really only minor alterations that are going to occur. And it's really more to do with movement of water, just to concentrate or dilute the urine as needed. This is just zooming in on the filtration membrane between the glomerular capillary and the visceral layer of the Bowman's capsule. And so you can see the red blood cells, so that helps kind of indicate that's our capillary there. And then you have the endothelial lining, all right? And then between the endothelial lining, we have our sort of filtration slits there. So again, size exclusion, there will be some charge exclusion there. This is in blue right here is the basement membrane. All right, and there's a little space over here between these, what they call these processes of the podocyte. If you recall, the podocytes are the specialized cells of the visceral portion of the Bowman's capsule. And so again, that's going to help us to exclude based on size and charge and so on. But ultimately what gets through: water, electrolytes, glucose, amino acids, vitamins, and bicarb, and so on. We'll talk more about those specifics in a little bit. So the forces involved in capillary filtration, we've gone over before. We just need to talk about the specific forces involved in glomerular capillaries, because there's a specialized type of capillary. So if you look here, you can see that we have our afferent arterial, right? Blood is moving towards the glomerulus. We have our glomerulus, and then there's the efferent arterial where the blood is leaving. And I told you it's an artery on either side. So it's the efferent over here, and it's the afferent over here. And what this really means for us is that we actually have a much higher pressure system. So this is BHP, stands for blood hydrostatic pressure. And so the hydrostatic pressure here is about 60 millimeters of mercury at the very beginning of this capillary system. So that's what's in the blood pushing out. That's our blood pressure. And down here at the efferent end is actually 59 millimeters of mercury. So it only really drops about 1 millimeter of mercury. Unlike the capillaries we've seen when we discussed earlier on in the semester, where you had a drop from about, let's say, 35 millimeters of mercury to about 10 or 15. So it's a much larger drop in pressure. The arterioles here are a much higher pressure system. We're dealing with 60, and it only drops to about 59. The oncotic pressure inside the blood goes from about 25, which is our typical. And if you recall the other capillaries from the beginning of the semester, the colloid osmotic pressure or the oncotic pressure didn't change. It stayed about 25 millimeters of mercury from the arterial side to the venule side. But in these capillaries, we're pushing out so much volume that it concentrates the proteins in the blood, and that increases oncotic pressure. So therefore, it starts out about 25 millimeters of mercury, which is typical, and then as volume is leaving and the proteins are concentrating because they can't get out, as they concentrate, the colloid osmotic pressure or the oncotic pressure rises. So it usually goes from about 25 up to about maybe 38 or 39 millimeters of mercury at the other end. So that pressure starts to rise, and remember, that's the pressure that's trying to pull water back in towards it. Okay. Now, that's two opposing forces, which is why we have here the colloid osmotic pressure. You'll see it says COP here is 32 inward. Again, that's the proteins trying to pull water in. 32 is the average, essentially, between our 25 and our 39 or so. So 32 is the average colloid osmotic pressure, so it's higher. And you'll see the capsule hydrostatic pressure, or capsular hydrostatic pressure, is 18. So that little space here, all right, does exert some pressure because as fluid gets in there, some of it's going to push back. And that's about 18 millimeters of mercury from both sides, from the arterial, afferent arterial side to the efferent arterial side. So that's again going to be trying to push fluid back in. So that's 18, and 32 is the pulling pressure in on average. So if you were to add those together, okay, you end up with about 50 millimeters of mercury trying to force fluid back into the arterial system, and you have essentially about 60 on the way out. All right, so the net filtration pressure is going to be about 10 millimeters of mercury, which is why NFP, net filtration pressure, is 10 millimeters of mercury out. So let's explain that again with my example here that kind of looks like a hamburger now that I'm looking at it. Anyway, it's a hamburger. Let's use green for lettuce because that's healthy. Okay, so here let's pretend this is our afferent side, and over here is the efferent side. All right, I drew it as one straight line just for simplicity's sake, and the bun here, this black line, represents our capsular space. Okay, not like the interstitial space of what we've discussed with prior capillaries. It's our capsular space. All right, now on the afferent side, hydrostatic pressures equal to about 60. All right, on the efferent side, hydrostatic pressure only drops to about 59. So we don't have a very big drop. All right, and again, that's an outward pressure, right? Outward pressure. And now the colloid osmotic pressure, the oncotic pressure, colloid osmotic pressure is equal to about 25 at the start. So that's trying to pull it back in. And at the efferent side of it, the colloid osmotic pressure can rise to, say, like 39 or so. So there's a rise. And again, that's because we're losing a lot of volume, so the proteins do concentrate, and that increases the colloid osmotic pressure. And that's going to be a pressure, an inward pressure. Right, so those are two opposing pressures. Hopefully you guys remember that. And from the prior example, they took the average of these two, which is about 32. Okay. Now, in this space here, in the capsular space, all right, between the two layers of the Bowman layer, the visceral and the parietal, you have a hydrostatic pressure of 18, and that is constant on both sides. All right, doesn't change. It's about 18 throughout. All right, and you have a colloid osmotic pressure of about 0 millimeters of mercury because protein doesn't get through and there's no proteins residing in that space. So there is no pulling action by proteins in that space there. All right. So when you take it into account, anything that's trying to force fluid back in, we can add together like 18 plus 25 or 18 plus 39, and we can get an idea of how we are filtering here at the arterial end versus filtering here at the efferent side of it. All right, so you can do that calculation and kind of see. And if you were to do that calculation, we could graph that out, and I'll show you that in just a minute. So again, this is just giving those numbers so you have it typed out for you: colloid osmotic pressure, glomerular, outward pressures, and so on. So that's all there for you, so you guys get an idea. The bottom is just talking about net filtration pressure. So the overall pressures, if we're sort of averaging some of them, would come out to about net outward of about 10 millimeters of mercury. Now, where this becomes a little more interesting, when we compare it to our regular capillary. So here's our typical capillary here, okay? So what I want to sort of point out to you guys here is that, or as a reminder, is you guys remember this horizontal line right here, right? This guy here, that represented our net oncotic force. And remember, the oncotic force didn't change. And it's mostly because we didn't lose a lot of volume in any particular capillary. And then over here, you see on the arterial side, the hydrostatic pressure was, in this case, the net hydrostatic pressure was right around like 28 in this example. And it drops steadily to the venule side, which, can drop to about, anywhere between 5 or 10 millimeters of mercury or whatever. But there's a pretty large drop in hydrostatic pressure. And over here, then, we have hydrostatic is greater than oncotic pressure, so therefore there's a lot of filtration taking place here. And then we have reabsorption taking place here. So hopefully this is just review for you guys. Over here, you'll notice this is actually what's happening at the glomerulus. This looks different. In fact, our net hydrostatic pressure is here. And you notice the drop is far less steep than we saw with our typical capillary. Okay. And again, this is the net hydrostatic pressure. So we've removed, or we've already subtracted about 18 millimeters of mercury from the hydrostatic pressure opposing the hydrostatic pressure from inside the blood vessel, which was 60. So that's why it's around 42. And then it only drops 1 millimeter of mercury across the whole length. So from 42 to about 41. And then over here, this represents the colloid osmotic pressure, which rises. Started out about 25 or so, and as it concentrated, it went up to at max about 39. The point here is this: that hydrostatic pressure is always greater than the oncotic pressure, even when it rises to a total of 39 here at the end, because the hydrostatic pressure at the efferent arterial side is still about 41, which means the hydrostatic pressure is always stronger than the oncotic pressure in this case, and so all you have is filtration. So that's a major point to make here, is that there is no reabsorption occurring in the glomerulus. The hydrostatic pressures are too high. So therefore, throughout the entire glomerulus, all you have is filtration. The GFR, all right, glomerular filtration rate, so amount of filtrate formed per minute by the two kidneys combined. So that's what I was talking about with that other cartoon from your notes. Okay, the GFR, which in that example was 120 mils per minute. Okay, again, that's total by the two kidneys combined, not per nephron. Okay, if we were to do it per nephron, it would be much, much smaller volumes. So again, I give you that range, about 100 to 125 mils per minute. If we were to just sort of extrapolate this, right, 100 to, let's say, 125 mils per minute, 125 mils is 0.125 liters per minute. So I could multiply that by 60 to get what it is, how many liters it is per hour. And I could multiply it again by 24 to find out how many liters per day are getting filtered. If you were to do that with 125, so 0.125 times 60 times 24, it would come out to 180 liters per day. So that's actually a substantial volume that you're filtering every single day in order to maintain homeostasis and get rid of waste. Now, the important thing is this. The GFR actually has to be very, very tightly regulated because remember the filtration that's occurring at that glomerulus is influenced by blood pressure. Remember, because we're filtering due to arterial pressures of 60 millimeters of mercury approximately. So if blood pressure were to go up, filtration would go up. Blood pressure goes down, glomerular filtration goes down because it's reflective of the pressures there, right? That's what the hydrostatic pressure represents. So therefore, we have to make sure we regulate because our blood pressure can change multiple times throughout the day, and the GFR has to remain very consistent. And it has to be regulated within a very sort of smaller range, if you will. And even when we're heavily exercising, that GFR won't change that dramatically. It will change, but not as dramatically as you'd think. And I want to kind of give you some examples here, though. But the purpose of sort of discussing this is to say that if the GFR is to get too high, meaning we're filtering a lot of volume, so a large volume per minute is getting into the nephron. That's not going to allow enough time to reabsorb everything. Because remember, the cells that line those tubules, they do have a max value and a rate with which they're optimal in. And if there's just too much going by, like in other words, if I had a conveyor belt and it just has way too many things zipping by, I can't do as efficient a job, and a lot of things are going to get past me. It's the same thing with the nephron tubule. The GFR is too fast, it's too high, too much higher volume. It's going to miss some stuff, and it's going to end up in the urine, and you end up with very large volumes of urine. On the other hand, if the GFR is too slow and it's kind of sludging through there, well, the cells can actually take more than they should, and sometimes we can even take wastes back that we don't want to take. And so that could also be detrimental to the kidney's function as well. And in point of fact, in clinical circumstances, generally if the GFR is going down, which is typical for kidney disease, we lose GFR, it tends to drop. And if it's prolonged, it's partly what we define as chronic kidney disease. So for example, a GFR that's less than 60 milliliters per minute, if that persists for greater than 3 months, that could be a partial definition for a chronic renal failure. Okay. So again, coming back to my point was that GFR has to be regulated. All right, and I want to give you a quick example here, but at the bottom you can see a little chart here where the amount filtered, about 180 liters per day. What's amazing about this, and I just want to point out as I circle it here, is what actually ends up being excreted a day. In other words, how much urine do we produce in a day? If we're filtering 180 liters, okay, how much of that actually gets turned into urine in a day? And the answer is about 1.5 liters per day, which means we reabsorb 178.5 liters a day. And so that's the point I was trying to make, is that even though you're filtering 180 liters, greater than 99% of it is going to be reabsorbed. That's the vast majority of what happens in the nephron, is reabsorption. So we're going to reabsorb most of that, and ultimately what we excrete is going to be just a very, a fraction of all that, and that's going to end up being about 1.5 liters in total. Okay, so again, this is to emphasize that reabsorption is one of the primary roles that takes place. And so my last example I want to give you guys is that if I let's say I have a 20% increase in my GFR. So let's start with a GFR that's 125 mils per minute. That's a typical GFR, okay? Now, let's say it increased by 20%. So I get a 20% increase in GFR. That's going to produce a GFR of about 150 mils per minute. You guys can do the math on that. 150 mils per minute. Now, hypothetically, let's just assume that my reabsorption rate is the same as what you see down here, which, if you can't see that, that reabsorption is 178.5 mils, okay, per day. So let's say that's fixed. Let's say that's our absorption rate, and that's the maximum that we can reabsorb. If I were to have a GFR that increased by 20%, and I didn't do anything to adjust it, it just went up to 150 mils, okay, what would actually be my total GFR per day? Well, you do the same calculation, right? 0.150. All right, that would be liters per minute times 60, which would give us how many per hour, times 24, which gives us per day. Okay, so if you do the calculation on that, you're going to see that we're going to end up with over 200 liters. Okay, in fact, we can do that calculation right now. I'll make sure I do my numbers, because make sure we get this correct. All right, so 0.150, all right, times 60 times 24, all right, comes out to 216 liters per day. Now, the point I'm making is this, is if I'm still reabsorbing at 178.5, okay, and my GFR went up 20% to 150 mils, now instead of 180 liters per minute, per day, which is typical, I'm now filtering 216 liters per day. And so if I want to know how much urine I'm going to produce now, that's 216 minus 178.5. Okay? In other words, it's almost 40 liters Okay, it's almost 40 liters of urine. That's not compatible with life. Okay, I can't urinate 40 liters. I would have to be drinking all day long just to offset that. Okay, and the reality is that's not actually what happens, right? We're going to adjust how much we reabsorb, and more importantly, we're going to control our GFR so it stays maintained. Okay, so we have ways or mechanisms with which we can adjust that and make sure that we maintain a GFR that's suitable for our reabsorption capacity. So regulation of glomerular filtration. GFR, all right, is controlled by adjusting glomerular blood pressure from moment to moment. So the way we're going to maintain it, again, if you remember that the glomerulus is surrounded by an arteriole, an afferent, and an efferent arteriole. So the GFR is controlled by three mechanisms: renal autoregulation, which is a myogenic response, which you guys have heard of. All right, that is contraction response to being stretched by those stretch receptors. So if there's a sudden surge in blood pressure, they'll contract or constrict to reduce flow into the glomerulus to help maintain the GFR. Then there's something called tubuloglomerular feedback, and that has to do with feedback from inside the nephron itself, telling the afferent or the efferent arteriole to constrict or relax to help adjust blood flow to that glomerulus. There is some sympathetic control, which is typical for the body when it's under duress, and then hormonal control, which we'll talk more about in lectures to come. So autoregulation, all right, myogenic mechanism. Again, stretch, a sudden surge in pressure causes the afferent arteriole to constrict, all right, and constriction of the afferent arteriole will block blood from being able to get into the glomerulus, which will help to maintain that GFR. Tubuloglomerular feedback, on the other hand, this has to do with the fact that there's a structure within the nephron tubules, specialized cells that will detect an increase or decrease in GFR. And in order to signal the afferent and efferent arteriole to constrict or relax in response to that increase or decrease in GFR, there's something called the juxtaglomerular apparatus. These specialized groupings of cells that help to create the appropriate response. So here in this cartoon, what I want you to see is this structure right here. All right. That structure right there, that's our corpuscle. You can see the glomerulus, you can see the Bowman's capsule. This over here would be the proximal convoluted tubule. This over here, cut away, is actually the part of the distal convoluted tubule. So the reality is, when I showed you that cartoon with the nephron all laid out, and you have the proximal convoluted tubule, and then the loop of Henle and the distal actually comes back right where the ascending portion of the loop of Henle. Here we go. Ascending loop of Henle becomes the distal convoluted tubule, actually comes back towards the corpuscle. So I'm going to give you an accurate drawing really quick. Is if this is the corpuscle, that's the Bowman's capsule. Here is the proximal convoluted tubule. All right, then the loop of Henle goes back. All right, so this was the loop of Henle, goes back here, and then we have over here, and then here's this is the collecting duct. So this is the distal convoluted tubule. So in reality is, and here's the proximal convoluted tubule. The reality is that this corpuscle, all right, is located very close to the initial portion of the distal convoluted tubule. All right. In fact, that's why we call it juxtaglomerular apparatus. It's right next to the glomerulus. And as you can see in this cartoon over here, this is the initial portion of the distal convoluted tubule, which I'm circling in red. And these specialized cells right here, which are important, are called the macula densa. Right? The macula densa. And so those specialized cells is where this all starts, actually. So what happens is, if, let's say, for an example, GFR is increased, that means there's increased flow going through those tubule lumen. What happens is that leaves less time for reabsorption, based on what I told you guys previously. Less time for reabsorption means more solutes are going to reach further along the tubules. So these macula densa cells, actually, what they're monitoring is sodium chloride levels. So if the GFR goes up, that means less reabsorption of sodium chloride. That means more sodium chloride reaches the macula densa. The macula densa senses this increase in sodium chloride levels. And what it does from there is it signals these cells over here, called granular cells or juxtaglomerular cells, and these mesangial cells, which are specialized types of smooth muscle, And what happens is, this is the, by the way, this is the afferent arteriole, and over here this is the efferent, right? If there's an increase in GFR, so there's increase in sodium chloride that reaches the macula densa, that means the GFR is elevated. So what it does is it signals these granular cells over here to constrict. That reduces the flow going into the glomerulus and thus reduces the GFR and restabilizes our GFR, thus our flow. Okay. What it can also do is it can relax the efferent arteriole. So constrict the afferent and relax the efferent, so that way blood is more likely to move out of the glomerulus and into the efferent arteriole by reducing resistance. Okay. So I dilate the efferent arteriole. That'll also help offset the pressures in the glomerulus. So again, you're going to see that there's going to be a toggling between constriction of one and relaxation of the other to offset this GFR. The opposite is true. If the GFR goes down, that means more reabsorption can take place. That means less sodium chloride makes it to the macula densa. That drop in sodium chloride sensed by the macula densa means that it's going to signal the granular cells, and when it does that, the afferent will actually dilate, so that way flow increases, while the efferent would constrict, and constriction of the efferent again would build up backward pressure into the glomerulus, therefore increasing the amount being filtered, so that way the GFR would rise. And the mesangial cells, they can play a role in ultimately relaxing or constricting the capillaries or the glomerulus, therefore increasing or decreasing surface area for filtration. In terms of the sympathetic nerve control, the sympathetic nerve fibers tend to constrict the afferent arteriolar smooth muscle, and so that's going to constrict the afferent arteriole. And again, if you recall from sympathetic stimulation, that we tend to turn off urination, right? So during, say, exercise or something like that. But generally, these are less potent under normal circumstances than our autoregulatory mechanisms. But under, say, strenuous exercise, okay, like a marathon runner or something like that, or acute conditions like circulatory shock where our blood pressure is really dropped, this will actually strongly constrict the afferent arteriole. In exercise, it's helpful because it's going to help shut off urination during fight or flight response, as well as to help increase blood pressure because we're maybe sweating a lot and losing a lot of volume, so it helps us maintain volume. Same thing for circulatory shock. It's going to constrict. Now, it may seem counterintuitive because if we have a low blood pressure already, that means the GFR might be dropping, but this is really an acute emergency sort of situation. So when it constricts there, yes, the GFR will drop, but the purpose of that is to not let too much get filtered so that we don't lose volume when we already have a very, very low blood pressure to begin with. So this is more of a life-saving measure. Again, just to reiterate, reabsorption, moving substances out of the tubular lumen into the capillaries, secretion going from the capillaries into the tubular lumen. A good rule of thumb to kind of follow with this when it comes to the substances that get filtered. Ones that get reabsorbed are generally not secreted, and ones that are secreted are generally not reabsorbed. There are exceptions to all these rules, of course, but that's a sort of a general rule to follow. So if we're going to talk more specifically about the cell types and how they actually move substances in or out of the nephron, all right, we have to kind of just quickly go over how substances move across cell membranes, right? So you have diffusion, transcellular or paracellular, which means around the cell, and this will require any energy, just gradients. And water primarily is reabsorbed via osmotic gradient, but there will be exceptions to that, and we'll talk about that too. Primary active transport, just to reiterate, needs ATP directly, and so things like calcium ATPase, proton ATPase, or the sodium potassium ATPase, or the sodium potassium pump, all will directly cleave phosphates in order to use that energy to move substances against their gradient. Secondary active transport uses the gradients that have been established by those primary active transporters to move things against their gradient. So if it moves, let's say sodium moves down its gradient and pulls something along with it into the cell, we refer to that as a symporter or a co-transporter. If it's sodium, let's say, moves into the cell but then pumps something out of the cell, like let's say a hydrogen ion, we refer to that as an antiporter or an exchanger. So I want to talk specifically now about the proximal convoluted tubule. So we're going to talk about each section individually, the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, because each section functions a little bit differently in terms of how it reabsorbs, what it reabsorbs, and how it secretes and what it secretes. So the proximal convoluted tubule reabsorbs about 2/3 of the glomerular filtrate. So 2/3 of that 120 mils per minute or so, that's going to be reabsorbed right away in the proximal convoluted tubule. This is driven by the sodium-potassium pump. So it's an important pump that has to maintain a gradient so that things can get moved into the cell. So again, we're going to be seeing things like symporters or co-transporters and exchangers. So you can see down in the bottom right-hand corner, there's a little cartoon of the nephron. And so what I want to do is just draw your attention to the segment which we'll be talking about, which is right here. This is the proximal convoluted tubule. And so what I want to do for you now is just draw some of the cells that would be lining the proximal convoluted tubule. So we'll draw a couple of them right here on one side, we'll draw a couple on the other side. And what I like to also put here on the outside of that proximal convoluted tubule would be the peritubular capillaries. I'm just going to draw these two red lines on either side to represent that peritubular capillary. And so our flow of filtrate coming from the corpuscle, say, is in this direction. All right, so that would be the inside or the lumen of the nephron. So that's the lumen. And these would be the capillaries. And so what's happening here, all right, is we're getting about approximately 2/3 reabsorption of the filtrate that's going to take place in the proximal convoluted tubule. Primarily 2/3 really of the sodium and the water will be reabsorbed here. And you'll see that some of the solutes will have differences in how much gets reabsorbed at this point. So let's first talk about sodium. So first off, in order for sodium to be taken in by these cells that line the proximal convoluted tubule, there needs to be a gradient. So I'm going to draw a little green box over here on what we call the basal side. All right, the basal side of the cell, which is facing the peritubular capillaries. All right, that little green box represents, so that's my green box here, that represents the sodium potassium pump. All right, a little positive there too. So that's the sodium potassium pump. And so if you guys remember, what that's going to do is that's going to pump sodium out of the cell in exchange for potassium inside the cell. Ultimately, the goal here is that the sodium levels inside the cell will be very, very low. Thus, any sodium that is actually located here in the lumen that's gotten filtered is going to want to make its way into the cell down its concentration gradient. And in fact, we're going to use the energy or the ATP that's required to power that sodium potassium pump to create a gradient so that sodium, that potential energy of sodium inside the lumen, will then help to drive in some of these other solutes. So for example, we're going to have on the apical side is the side that faces the, the lumen of the nephron, we're going to have co-transporters. So I do that co-transporter in purple there. And it's going to drive some solutes into the cell. And some of those solutes are glucose, sorry about my handwriting, amino acids, those are two A's there, amino acids, okay. Other ones like phosphates. So these can be all co-transported in with sodium. And so sodium helps to drive them in. And what you're going to find is that glucose is about 100% under normal conditions reabsorbed in the proximal convoluted tubule. The amino acids are about 100% reabsorbed in the proximal convoluted tubule. And phosphates, they vary a little bit more. It could be 80 or 85% or so. And there's one more I want to show you. There's going to be another co-transporter, or I should say, excuse me, an exchanger. So I'm just going to draw it on the cell over here, but understand any one cell can have multiple co-transporters or exchangers, but I'm going to draw it over here just to have a little space to work with. And so sodium, which we'll draw back in green over here to keep it consistent. So sodium, again, is going to want to move into the cell, down its concentration gradient. And it's going to move in exchange for, over here, it's going to be an exchange for, so we're secreting at this point. We're going to secrete a proton, or that's a hydrogen ion. We're going to secrete a proton. And now that proton, although it's being secreted here in the proximal convoluted tubule, will not ultimately end up in the urine. It will not be excreted, even though it is being secreted here. We're going to need that proton ultimately to pull in bicarb, which has been filtered, HCO3 minus. And we'll talk about the mechanism with which it does that on an acid-base lecture. But the bicarb is going to get reabsorbed, and we're going to reabsorb about 90% of that bicarb in the proximal convoluted tubule. And the mechanism is basically we're going to take advantage of this hydrogen ion that's being secreted into the lumen, and we'll ultimately be able to reabsorb the bicarb as well. So this slide is really just reviewing again the reabsorption that takes place in the proximal convoluted tubule. And so you can see on the slide that in the cartoon that we have the tubular lumen over here, which I'll circle for you in blue. That's the tubular lumen, so that's the inside of the nephron. And then over here is the interstitial fluid, which is where the peritubular capillaries would be located. And these would be, again, the cells that line the nephron. And so you can see on the apical side over here, you can see a glucose sodium co-transporting glucose and sodium co-transporting amino acids, just like in the cartoon that I drew on the previous slide. So I mean, yes, some of these transporters will have specific name, sodium glucose transporters, for example. Once they get inside the cell, all right, glucose can then leave the basal side of the cell through glucose facilitated transport and into the interstitial fluid, ultimately to return to the blood. And then here's our pump, right? So here's our sodium-potassium pump, which creates the gradient for sodium over here to be driven into the cell. Amino acids, again, can be moved through facilitated transport out of the cell on the basal side. And then over here, it's just showing you the sodium-hydrogen exchanger, or NHE here. And so sodium moves in, the proton or the hydrogen ion gets moved into the lumen, and ultimately we'll talk about how that helps us to reabsorb the bicarb. What we didn't talk about is the chloride. Chloride ions generally follow the positive sodium ions by electrical attraction, and they will move usually passively in between the cells and sometimes directly through the cells through some special transporters as well. So in terms of reabsorption, just as a sort of summary here, each day the kidneys reduce 180 liters of that GFR to about 1.5 liters of urine. In other words, although there's 180 liters moving through the nephrons every single day, ultimately what's produced in the end is only 1.5 liters of urine, which means primarily we're reabsorbing. Okay, so we've talked about that on earlier slides. So 2/3 of the water in the filtrates reabsorbed in the proximal convoluted tubule, and the water, the way it's reabsorbed, is just through osmosis. So what happens is these are you can kind of view these cells that line the proximal convoluted tubule as being fairly leaky. And so what happens is as the solutes are being actively reabsorbed, they're going to get pulled in first, and then the water will follow with it. And then finally with secretion, we'll talk more about secretion in the next lecture, but secretion, this is where ultimately things are going to move from the capillary or from inside the cells that line the nephron and be moved into the lumen of the nephron. So we refer to that as secretion. And there's several purposes for secretion. And in the proximal convoluted tubule, that's going to help us with acid-base balance, so that hydrogen ion that was secreted will help us recover bicarb, which again, bicarb is a buffer that will be used for acid-base regulation. Waste removal. So there will be some secretion of things like urea, uric acid, bile acids, ammonia, some other waste products. And then finally, clearance of drugs and contaminants. Basically things like morphine, penicillin, and aspirin, just to name a few of the medications that would ultimately be cleared by the kidneys, and some drugs that, might need to be taken multiple times are dosed according to the renal clearance based on the kidney's ability to get rid of that drug. And so you try to dose it accordingly so that you can keep an optimal level of medication in the person's blood. In some cases, we would call that like a plateau level, which would be the therapeutic dose. So it's really a matter of adjusting inputs relative to the kidney's output or excretion of that drug. All right, so we will talk more to that on the next lecture. If you have any questions, please email me.